Yes. Good afternoon, everyone, and thank you for joining us again in the fifth webinar of the African Society for Craniofacial, Dental and Craniofacial Genetics. We are honored and excited today to have Emeritus Professor Anwar Suleman Mall that will give us a talk about the changing face of the biological science in the 21st century. Professor Anwar Suleman Mall is a renowned expert in physiology, biochemistry, and surgical sciences, holding a PhD from the University of Newcastle upon Tyne. Professor Mall has made significant contribution to the field, particularly in the role of mucus in health and disease. His distinguished career includes training numerous honors, master's and PhD students, and presenting at approximately 30 international conferences. Professor Mall has held various prestigious positions, including Emeritus Professor and Senior Scholar in the Department of Surgery at UCT, Acting Deputy Vice Chancellor for Student Affairs and Transformation, and Assistant Dean of Student Matters. His accolades include the Golden Key Award, the Oppenheimer Award for Research at the University of Oxford, and the Distinguished Teacher Award. As a National Research Foundation rated scientist, Professor Mall's insight and experiences are invaluable, indeed. So, as usual, any comment, question, or point to raise at the discussion, you can pop it during the presentation at the chat, and we can discuss it or address it later on. So without further ado, Professor, you may start.
Thank you very much. Thank you, Salma, for that introduction. So welcome, everybody, and I hope you can hear me. Please shout if you can't. So I'm going to talk about this changing face of the biological sciences in this 21st century. And I think it's a very relevant topic for us to be able to understand, the very foundation of whatever we do, whether it's clinical medicine, clinical dentistry, dental research. We have to actually keep up with what is going on in terms of the broader scheme of things, in terms of what people are thinking. So I'm going to just give you a little overview of what has happened in the past and where the changes are taking place. Now, how do I change? Sorry, how do I change? It's not.
Yeah, I think just the arrows, use the arrows on the right if you're
Okay, that way, right.
Yes.
Okay. So it all started with this man called Charles Darwin in 1859, or more than 160 years ago. And Darwin, he gave us the theory of evolution, one of the most beautiful theories, which has survived time for over 160 years and hasn't as yet been given a serious challenge. And what Darwin told us is that we all are united in our singularity and diversity from one single cell. Can you see my arrow? Can you see my arrow here? From one single cell right up to so many different species up to us right here. And that is how we came about. And obviously, he said there was a hereditary factor that was transmitted from generation to generation to allow this speciation and to allow for this diversity. And what the problem was with Darwin was that he didn't know what the nature of the hereditary material was that was passed on from one generation to another. So the surprise thing is that not very far from Darwin lived in Kent in England, and in Austria in about 1856, as part of the search for the hereditary factor, this monk, Gregor Johann Mendel, he confirmed that there were units of heredity that were passed on from generation to generation, parents passing on this unit to their children. And he was a scientist, he was a friar in Czechoslovakia. And what he did was he worked with 7 characteristics of pea plants: plant height, pod shape, seed shape, and color. And he showed that when a yellow pea and a green pea were bred together, their offspring plant was always yellow. However, in the next generation of plants, the green peas reappeared at a ratio of 1:3. So to explain this, Mendel coined the term recessive and dominant, which we still use today in genetics. And his pea plant experiments conducted between 1856 and 1863 established many of the rules of hereditary, now referred to as the laws of Mendelian inheritance. And not long after that, this man, his name was Johann Friedrich Miescher. He was a Swiss physician and biologist. He took white cells with big nuclei from pus in bandages and sperm from salmon and extracted a macromolecular compound rich in phosphorus, and he named it nuclein. And then in 1889, it was called nucleic acids. And nucleic acids are today what we call DNA and RNA. But this search for hereditary material went on for almost 100 years. And these molecules, the what we call DNA and RNA today, were the last to be isolated. This is a lovely book to read. It's a history of molecular biology, and I bought it for only $1. Okay. Now, then about almost 100 years later, in 1944, a physicist, a Nobel laureate, he writes a book called What is Life? And the only thing I'm going to tell you about this book is that Francis Crick read it. I'm going to talk about Francis Crick. But he looked at life from the point of view of physics, and he wasn't very far off the mark. But the man that we should actually all admire was this man, Oswald Avery, at the Rockefeller Institute, because in 1994, he did the definitive experiment. He took two different types of bacteria, one that had a gene for a disease, the other one that didn't have a gene for a disease, and he crossed them, and when they crossed over, the ones that didn't have the disease became ill. So he said inheritable transformations occur in a strain of pneumonia bacteria when they are mixed with DNA from a different strain. And in that very year, when Schrödinger published What is Life, this is what Oswald Avery found. He did the definitive experiment, and he says the chromosome is a packaged and organized structure containing most of the DNA of a living cell. And most of our cells, the eukaryotic cells as we know them, have a set of chromosomes, 46 in human. So you've got 46 chromosomes. You've inherited 23 from your dad and 23 from your mom. Forgive me, I know you know all this, but I'm just setting the scene for the debate. And so we reach our climax. The climax was that in 1953, these two guys, Francis Crick and James Watson, actually worked out the structure of this hereditary unit, and it was called DNA. And Francis Crick said that he read What is Life, and he got some ideas. Schrodinger's What is Life. And he said biology has become very much about information concealed in the sequence of genes and proteins. And they worked it out in this pub. They worked it out in a pub because they used to go to a pub every day. And I went to this pub and I asked the publican there, I said, where do you think they were sitting when they worked out the structure of the DNA? And he says, no, apparently, historically, we know that when they came in, they sat anywhere as long as they got their beer. So that was a nice little piece of history. And so James Watson, Francis Crick, and Maurice Wilkins of London, who was collaborating with them, they won the Nobel Prize. But it wasn't entirely a happy story. It was a bit of a sad story because the person who actually did the X-rays of DNA and found that it was a double helical structure was Rosalind Franklin. She unfortunately died before they worked out the structure. She was very controversial. She was responsible for much of the research and discovery work that led to the understanding of the structure of DNA. But she died at the age of 37, And they don't actually give the Nobel posthumously. Okay. And that was very sad. And I did my own crying when I stood outside a London theatre in the West End when my favorite actress, Nicole Kidman, was playing Rosalind Franklin. And it was called Photograph 51, the play was called. But the starting price of the tickets was £60, and you can imagine what that would be in rands. So that was unfortunate. So from all this came out something, a central dogma, which all of you have been taught in basic biology, that DNA can make a copy of itself, it can transcribe into RNA, and RNA can translate into protein. And that became the central dogma of protein synthesis. The other name for it is the information theory of life, which has dominated thinking since the mid 19th century. Okay. Now, there's been recently a lot of rethinking about this whole thing. Is this right? There's people like Nick Lane. He works at the University College London. He's a biochemist. And he's written several books, several brilliant books about why he thinks that we have made progress in biology, but it's not enough. And he says that we are caught at the stage of this information theory, and we can't seem to move from there, which is a very interesting point he's made. And his main opposition, I've just chosen these two, there are others. The main opposition is the very famous Richard Dawkins, who you might all know as the militant atheist of the world. But all this has raised a new question. What is life? It's one of the biggest questions in science. How do inert atoms and molecules found in rocks are transformed into running, jumping, flying, navigating, swimming, growing, loving, hating, lusting, fearing, thinking, laughing, crying, living stuff? That's what people want to know. How come we are alive and all so much around us? There's so much life around us, and it came from inanimate matter. People say we came from stardust. People say we're just made of atoms. But so is the desk I'm sitting at. It's made of atoms, but the desk is not alive, and I'm made of atoms, and I'm alive. So these are the questions that are actually bugging the scientists of the world. Now, Dawkins wrote this book, The Selfish Gene, in 1978. And funnily enough, I was talking about it to somebody at lunch today, and that person was raving about this book. So I told him, well, what Dawkins has done, he knew that Darwin didn't know what the hereditary factor was. And he now had evidence in the 1970s, of course, lots of evidence that the hereditary factor is a gene, which is DNA, right? And he wanted to synthesize, he wanted to integrate genetic theory with Darwin's views, because he said they just complemented each other so beautifully. So it became the gene-centered view of information theory. And he says in his book, in the beginning was simplicity. Simplicity changed gradually into complexity through evolution and natural selection. Unordered atoms grouped themselves into complex patterns that eventually led to people. He was absolutely quite sure about it. And all these statements are being challenged today. But at that time, I must tell you, it was a watershed book. Okay. So what did Darwin think? How did life come about? How did it originate? Darwin said that there was the first replicator. Like we talk about replicator today, we mean DNA. We know it replicates. That means it can make a copy of itself. Now, that first replicator was an ancestor of DNA. At some point in the history of the universe, a particularly remarkable molecule formed that had the extraordinary property of being able to make a copy of itself, an extremely improbable event. And it only happened once. It was an unlikely sort of accident to happen once. Now, I want you to put this in the back of your minds, because this is being challenged today. It made many copies of itself. Some survived and others not. Natural selection favored the most stable ones, and they gave us the forerunner of the gene. And then he made this very bombastic statement: the gene is the unit for natural selection. So if you are successfully alive, you have been selected out based on your genes. If your genes are faulty, you don't survive. So that was Dawkins' theory. Okay. Now, then he went on to say something even more controversial, which at that time was accepted, I must say, but it's controversial today. These replicators, this DNA, they form cells. So he says replicators began not merely to exist, but to construct for themselves containers. So first they were out in the open. Now they found themselves inside a living cell. And this living cell is a survival machine for themselves to live in. Today, 4 billion years later, they are our DNA, the genes, the past masters of the survival arts. So what he is actually telling us is these genes rule our lives. So no matter who you might be a celebrated footballer, you might be a world famous singer. The genes are not worried about all that. All that the genes want you to do is to be happy, so you reproduce and you pass them on to a next generation. So they are our masters. And all these thoughts today are being unpacked. But I just wanted to show you, um, so underneath this, if you can't see the slide, I've got atoms, physics. So they say all matter in the universe is made of atoms. And when atoms join together, they actually form molecules, like two hydrogens will form an oxygen. Now, that is chemistry. That is chemistry. And then these molecules can grow into something larger. So the chemical bonds can grow and grow, and you can make macromolecules, huge molecules like proteins, lipids, nucleic acids, carbohydrates. These are organic compounds. Now, these are the compounds that make us and make the living things, make your cat and make the ants and make everything. And now, if you arrange them in a certain way, you get a cell. Aha. Now, this is where the issue lies. When you asked what is life, or when you are asked, how come that these atoms are not alive? Water or other molecules, macromolecules, are not living things, they're inanimate things. And yet, these inanimate things in a particular arrangement give you a cell, and the cell has a new property. It is alive. That means the whole is more than the sum of its parts. None of these are alive, but if you arrange them in a certain way and form a cell, which I have here as a simple diagram, then something new has emerged, a new property. And how does that come about? And that's the property of life. The whole is more than the sum of its parts. And that's the question that is bugging scientists, okay? And that went even further. From atoms, you make molecules, you make cells. And once you make cells, you know that all forms of life are made of trillions and trillions of cells. But first, cells have to become join with other cells and become tissues. And then you'll get organs, organ systems, and an organism, you and your cat. And if you're a human being, you have a mind and consciousness, and you are part of a society, a culture. You can't live on your own. And again, that culture has given us so much. Exchange of thinking at any level has given us the humanities, economics, literature, philosophy, and political systems, religions, and all that, right? So we live in societies because we're successful and we live altruistically. So if you're selfish, you won't last long. If you're generous and if you're altruistic and if you care for the environment, if you care for your fellow human beings, if you care for animals, then this system then controls what goes on at the bottom. Okay, but I don't want to get into that now. I just want to actually give you an idea of what the thinking is like. But anyway, the gene story continued. So Dawkins wrote his book in 1978, and then the Human Genome Project came about. And the Human Genome Project was an international scientific research project in about the year 2000, with the goal of identifying, mapping, and sequencing all of the genes of the human genome. And you know, today we've mapped the entire human genome, right? And that genome is thought to be the blueprint of life. And one of the first books was by Francis Collins, right? Who was one of the scientists who mapped the human genome. And he says he found God, and he wrote this book, The Language of God, because he now knew what God's language was when he created us. Now, let's see. Let's come to today. So now this year, there's just been a book called How Life Works. It's a very thick book. It's written by this man, Philip Ball. Philip Ball was a physicist, but he did a lot of work in biology. He spent a lot of time at Harvard Medical School. Now, he says when the Human Genome Project was announced, Bill Clinton was then the president of the United States, and he made a speech. Today, we are learning the language in which God created life. Okay, so when Bill Clinton took to a White House lectern 20 years ago to announce that the human genome sequence that we have mapped and completed is a breakthrough as the most important, most wondrous map ever produced by mankind, or humankind, sorry. The scientific achievement was placed on par with the moon landings. And there was a promise made. The promise was it would revolutionize the diagnosis, the prevention, and the treatment of most, if not all, human diseases. So that was the promise. So this was a blueprint. And if you knew your genome, if you mapped it, then you had your book of life. Okay. And now the search began for which genes may be responsible for humanity's illnesses. Yet when scientists, now this is interesting, now this is where the changes start coming. When scientists investigated which genes are linked to human diseases, they were met with a surprise. After comparing huge samples of human DNA to find whether certain genes led to certain illnesses, they found that many unexpected sections of the genome were involved in the development of human disease. So if I had a brain tumor, thank heaven I don't, but if I had a brain tumor, and if they mapped my genome to see which gene has caused my brain tumor, they would find, and I'm just giving you a ridiculous example, they would find that it's a gene that secretes acid in the stomach. And that doesn't gel. So they were getting pretty worried that they were not nailing the genes that were responsible for the actual diseases. And then the question of what is life remained. And here's another lovely book by Paul Nurse. If you're interested, you can actually read it. He's also a Nobel laureate from Oxford University. Okay. So this man, Dennis Noble, he's one of the people who said, no, no, no. And he's a friend of Richard Dawkins. They work together. He said, Richard is wrong. Richard Dawkins is wrong. This idea of genes making cells, of genes making copy of themselves, he says, and everything relies on genes, can't be. He says, we call it a blueprint, but we should actually look at it as a musical score. And he wrote this book, The Music of Life, which he actually sent me a copy as a gift, which I was very thrilled about because I don't know him. Now, he says it's not a blueprint. The blueprint metaphor was not the right way to think about the genome. And he gives an example. He says an admirer once said to the Russian violinist, Yasa Heifetz, your violin has such a beautiful tone. And Yasa Heifetz took his violin and put it to his ear and he says, I don't know what you're hearing, but I can't hear anything. Why do you say it has a beautiful tone? The moral of the story was, you need an orchestra to produce the music. The musical score by itself is not the cause of music and is not enough, right? And he said, Dennis Noble said, take your entire genome from your cells and put it in a test tube. It won't do anything. It won't do anything. So the genome is actually being quite overrated, okay? So, okay, I've done this slide. Then Nick Lane comes along and he says, I challenge this information theory of Richard Dawkins and company. He says biology has been dominated by this information, the power of genes. But what difference between the information content of a living cell and one that died a moment ago? Okay, because if I suddenly die now, somebody will come and say, well, all his parts are still there. What left him? What is missing? So this gene theory, they said if he's got genes and genes make life, why aren't the genes giving him life? It creates a problem in the trajectory of evolution and the long delays between abrupt changes, because bacteria and archaea, they appeared on this planet 4 billion years ago, but the cells that make us, these were unicellular organisms, obviously, but the cells that make us, the eukaryotic cell, right, which led to the multicellularity that we display today, or your cat displays, or an ant displays, or a bird displays. So you had to turn into a eukaryotic cell, okay? This is what Nick says. And genes had nothing to do with it. And he says, really, Dr. Dawkins, replicators give rise to cells. He says, let me tell you something. Anthony van Leeuwenhoek in the 7th century first described a cell. And we have dissected the cell to bits and know all its working parts. And yet underneath it all, we are barely any closer to understanding what breathes life into these flicks of matter. How did they first emerge from the sterile, inorganic, inanimate earth? What forces coordinate their exquisite behavior? Do they experience any sort of feelings? So he says there is a paradox at the origin of life. To a first approximation, biology is understood in terms of information networks and control systems, right? So this view that genes were first as replicators and they make copies of itself, it generates a paradox at the origin of life and raises the question, where does all this information they have to make cells and to make us come from? So they say it comes from natural selection, but I mean, that doesn't make sense. So Nick Lane proposed that this is what happened. And he wrote this brilliant book, The Vital Question, right? And there he opposes this view of the genes being the first things that originated on this earth. He says all complex life on earth share a common ancestor, a cell that arose from simple bacterial progenitors on one occasion in 4 billion years. This common ancestor was already a very complex cell, and it passed this complexity and sophistication to its descendants, that's us, the trees, and the bees. All complex life shares an astonishing catalogue of elaborate traits, from sex to cell suicide to old age, none of which is seen in a comparable form in bacteria. So our cells, the father of our cells, when it appeared on this earth, was a very sophisticated cell. If this complexity is not in bacteria, which arose 4 billion years ago, and it is there in our ancestral cell 2 billion years ago, how could our ancestor, the eukaryotic cell, have arisen from the bacteria, which is a prokaryote? Did evolution and natural selection not occur between these two types of cell? Did a bacteria become a eukaryote, one simple and the other complex? What are the intermediate types between simplicity and complexity? So he's asking these questions, and he says there is a black hole at the heart of biology, because the first living cell was a bacterium or an archaeal cell, which is a plant. And he says that was the last universal common ancestor called Lucha. And then 2 billion years later, we get the last eukaryotic cell ancestor, the cell that is our ancestor, the father of multicellular life. Okay, so there is a gulf between Lucha and Leka by about 2 billion years. There is no indication that archaea or bacteria evolved into eukaryotes. People say, oh, from the very beginning, there was evolution of this, that, and the other. Where are the intermediate steps? He says they're not there. He's a very, very controversial man, this, I must tell you. Okay. But he writes beautifully. This is his theory. He says, yes, 4 billion years ago, Lucha appeared. It was the father of bacteria and archaea. So these two branches existed. And they were unicellular organisms, but they were simple organisms. But they were not unintelligent. They knew how to approach food. They knew how to move away from danger. But something very strange happened, ladies and gentlemen. Along the way, 2 billion years later, a bacterium entered an archaeal cell. So there was an invasion by one cell type into another cell type. And now you've got a more complex cell, and that is Lecha, our ancestor, Leca. And he says, we from there, all the diversity of life that you see outside bacteria, this is the cell. And it all happened by lateral transfer. Now, that new cell that invaded this host cell, right? The host cell was very upset and said, why are you coming into my territory? Get the hell out of here. And this new cell said, look, if you let me stay, if you give me this protection, I will be your powerhouse. Not ESCOM, a better powerhouse than ESCOM. Can we think of a good reason why one cell getting inside another cell should transform the prospects of prokaryotes, unleashing the potential of eukaryotic complexity? Yes, says Nick Lane, there is a compelling reason. It relates to energy. And this new cell that invaded that host cell became our cell's mitochondria. Okay, and there it sits. There's a lot of other organelles in the cell, as you know, but there is the one that was an original bacterium, and that became the powerhouse of our cells. It became the mitochondria, and it provides this whole cell with energy, and it's through that we became multicellular. And then from multicellular, we became what we are, whether we are humans or dogs or cats or birds or crocodiles or anything. Okay. Folks, are you with me? Hello?
Yes, yes, Prof, we can hear you clearly.
Okay. Right. And so Nick Lane, when he formulated this theory, he wrote this brilliant book, Power, Sex, and Suicide, Mitochondria and the Meaning of Life. If you're interested, you can read it. It's a lovely book, okay? And then there were other people who said, we must move away to the cell, we must move to the cell-centric perspective. DNA by itself, the genome by itself is inert. It needs a cell to work its wonders. And that the cell is always working with its environment. So DNA can only function in a cell, and it only functions when the cell cytoplasm asks it to. When the cell says DNA code for this protein, then DNA complies. Otherwise, DNA is inert. As I told you, you take it out of your cell, put it in a test tube, it will do nothing. So Dawkins' idea that this father of replicators, or this ancestor of replicators, formed in the original swamps by itself and replicated, is completely and utterly wrong. And I hope you're appreciating how brilliantly science progresses. When Dawkins wrote his book in 1978, it was a watershed book in biology. Even the sociologists, anthropologists were quoting from it and said, now we're finding out what life's about. In fact, the very meaning of existence is coming out of this. And it sort of, it influenced all fields of thinking. And now, so many years later, people are saying, no, life didn't start like that. Life started differently. But first you had to make the components of what makes a cell. So what is Nick Lane's alternative to Dawkins' gene theory? How did life begin? He says the origin of life was driven by energy flux, and proton gradients were central to the emergence of cells. Now, you know what? This man loves proton gradients. That means a high concentration of protons on one side of a wall or on one side of a membrane, and a low concentration on the other side of the membrane. He says that is the very essence of life. And I'll tell you a little more about what he meant. So what he says is that the first organic, remember, we are organic, we are made of organic carbon-based molecules. The first organic life-forming carbon-based molecules, he says, the important thing to know is all life on this earth comes from carbon dioxide and hydrogen, right? We come from the basic gases of the very early universe. Now, the oceans were acidic and they had a lot of carbon dioxide, okay? And they were separated. Remember the barrier? They were separated from the alkaline fluids rich in hydrogen by a thin inorganic wall containing iron minerals, iron sulfide minerals in the micropores. So here's a diagram. Here is the hot vent in which is a lot of hydrogen. And on the other side of the wall is the ocean, which is alkaline. And in between there is FES, which plays the role of an enzyme, okay? And he says at a particular pH, right, this hydrogen crossed over into the ocean and combined with carbon dioxide. And this is how we began. The first organic life-forming carbon-based molecule, HCHO, this one, CH2O formed. And he says, this wall, sorry, this wall between the ocean and the deep sea vent, the hot vent, right? This is the first proton gradient, and it's called chemiosmotic coupling. And FES is acting as an enzyme, right? The importance of a barrier. Other barriers we know today as the cell membrane, and then a proton gradient in chemiosmotic coupling, both at the beginning of life and in the first prokaryotes, the first forms of life, and eventually in us, the eukaryotes. These barriers are most important for life. Oh, okay, so what is it now? It's all happening in the mitochondria, that invader, that bacterial invader, right? Who now is the powerhouse of the cell. So we move from the cytoplasm into the mitochondrial matrix. And all of you must have agonized when you were students to learn the citric acid cycle. And we know that the food we eat is broken down in our cells and passed into the mitochondria in the form of glucose or fatty acid or whatever. And they come in there and they go through this cycle and they actually make ATP, and that ATP is the energy. This is what Nick Lane is saying, is that energy was important at the beginning of life, okay? But if we made from hydrogen carbon dioxide, how did all this form? How did all this form? And that's what Nick Lane tells us about, okay? But first he says, look at it. After the citric acid cycle, the electrons that are generated, they move along this inner mitochondrial membrane, okay? And there's a proton pump, which pumps out protons, which when goes into the ATPase, in that inner mitochondrial membrane, it makes ATP, and that's our energy. But I won't go too much into that. I want to actually get to the real stuff. Now, this is your central pathway of life. The very fact that you're sitting there listening to me, or I am speaking to you, we're consuming energy. And we are happy that in our mitochondria, this is moving in a clockwise direction, making ATP and everything else. Now, these intermediates here can also make amino acids. They can also make nucleic acids. So Nick Lane's central point is, first you had to have this before you could have amino acids and proteins, before you could have DNA and RNA, the nucleic acids. He said everything was made by these intermediates, but he still didn't tell us how these intermediates, which were first made, came to be. Okay, all I want to just say to you is this man here, Hans Krebs of Oxford, he worked this cycle out in the 1960s. And my first professor at UCT, Professor Wieland Gevers was his PhD student. Okay. Now, what we didn't know and what Nick showed us, that this cycle that moved this way to produce and give us ATP, right? Okay, can also in some parts move in reverse. Okay, and this is how life began. He says the minute you formed that first organic molecule, HCHO, which I showed you in the sea, right? That began to double and treble into all these intermediates, became more complex synthesis, became more complex. And as the citric acid cycle began to form, right, it worked in a reverse direction. Not as in our case at the moment, where it's working in the other direction to give you energy, it worked in this direction to give you growth. That means these are the first components of the cell. And remember, when they are made, they can make amino acids, which can make protein. They can make nucleic acids, which can make DNA and RNA. So for the cycle to go in reverse flux, right, in and out of the cycle, think of the roundabout, the traffic circle. Our multicellularity allows tissues to change their metabolic states in parallel. Nick Lane is saying that while you're sitting there, right, your liver cells might be going in that direction in the mitochondria, not the cells, the citric acid cycle, sorry, right, to give you energy. Whereas in some other tissue, which had a shortage of this, it's going in the other way to make these intermediates. Different tissue have different needs, some with high energy demands, others with specific biosynthetic requirements. Tissue like gonads, which require less oxygen, ferment sugars via glycolysis. So the biosynthetic mode of the reverse cycle is not unlike what happens in cancer cells. That's where his new theory of disease comes in, not with the DNA so much, although he says don't ignore DNA, it's very important, but he's got a new theory, right? So the forward cycle gives us energy and keeps us alive. The backward cycle gives us growth and gives us all these substances that can eventually make, so if you're short of amino acids and you want to make a protein, you can always come tapping to the reverse citric acid cycle. And it's built from quantities of hydrogen and carbon dioxide at the beginning of time. Okay, so disease. So the idea of a simple cycle just burning glucose persists. I mean, I used to teach this in biochemistry, the citric acid cycle. Students hated it, but when you explain it to them as to what it means and how energy is formed and how this keeps you alive, it keeps your brain working, keeps your organs working, it keeps every cell in your body working, and so it keeps you healthy. So that idea persists and it's fine. But now we are talking about the power of the metabolic cycles, which is being brought to bear on the underlying metabolic basis of age-related diseases, from cancer to diabetes and neurodegeneration. The simplistic notion is beginning to unravel, exposing the delicate symbiosis between tissues that underpins our health and lifespan. Okay, so we've got new explanations. So how do we age? If you've got hypoxia, if you've got infections, you've got inflammation, you've got mutations, all can alter the flow of food through the citric acid cycle, with a knock-on effect that switches on or off hundreds of thousands of genes, changing the stable epigenetic state of cells and tissues. Tissue function is strained, biosynthetic pathways falter, ATP synthesis declines, and the delicate web of symbiosis between tissues begins to fray, and so we age. Okay, so take cancer. Now we know cancer to be the dominant paradigm in cancer is that genetic mutations cause cancer. Yes, they do. If you have a mutation in a particular gene, it can start causing uncontrolled growth of your cells and you get cancer, okay? So these mutations lock cells into a pattern of uncontrolled growth, but that is not the whole story. People over 50 have 99 times the risk of people younger than 24. So Could it be that aging is the accumulation of mutations as we age? No. Our lives are too short to accumulate so many mutations to, to cause diseases. He says there is another cause, but he does concede that mutations in genes are important, okay? But he says we've become genetic determinists. Much of the research we've done based on this mutation cancer story failed to deliver on its promise of therapies. The death rate from malignant cancers hasn't changed since 1971, and we have spent billions on the problem. We know we have oncogenes and tumor suppressor genes, and they do mutate and they do drive cancers. But the context is far more important than the paradigm might apply. So there's another view. In cancer, he says these pathways that give us energy, they are become faulty. In cancer growth is the key. It's uncontrolled growth of cells. That's cancer. And growth needs more than ATP, not just ATP. The underlying fault in cancer is not a shortfall in ATP, but a shift in metabolic flux and gene activity towards a state favoring growth. And that is the reverse citric acid cycle. So cells must wire their metabolism to get the balance between ATP and everything else right. Okay, so what he's saying is the Krebs cycle, the citric acid cycle, is a two-pronged pathway in which one, if it goes one way, it gives you energy, okay? And then there's another pathway, as I showed you, the reverse pathway. So there's a yin and yang. And what we should be actually looking at, that in every cell in our body, there is this continuous movement this way and that way. But there has to be a balance between them. And the two forks join in the vicinity of succinate, right? Now, succinate is one of the intermediates in the cycle. It's a tipping point where citric acid cycle flux could go one way or the other. It could either go that way, or it could go from succinate to this way, depending on how much oxygen is available. So that succinate buildup happens in the absence of oxygen, which blocks the breakdown of a transcription factor, HIVF1 alpha. This binds to DNA and switches on hundreds of genes. Switched on genes encode proteins that drive growth, survival, and inflammation, which, if persist, can lead to cancer. So what he's saying is, start looking at these cycles. Are they balanced in the tissue? And start looking at the genes of the enzymes in these cycles. So the mitochondria is of almighty importance to understand cancer and other diseases, okay? Let's go, okay. Now, he says, if you take a tumor, you get one cell that transforms, which is a rogue cell. And then, but why are the altered or mutated genes of the transformed cell not always written into all of its descendants? So this cell goes mad. It starts just dividing, multiplying uncontrollably. But if you look at all the cells it's producing, it doesn't have the original mutations. The tumor cells are heterogeneous. And more importantly, normal tissue surrounding a tumor is always found to sometimes have tumor cells far away from the site of the tumor with no consequences. Taking cancer cells from a tumor microenvironment and implanting them in a normal cellular environment tends to halt their growth and often promotes programmed cell death. So cancer cells can be rewired, and the same genetic mutations are not invariably associated with the same cell fate. So he says the lesson is, older work has shown that transplanting the nucleus, which has your genome, from one cell type to another does not alter the phenotype of the recipient cell. Why? Because when you implant a cancer nucleus into a normal cell, the cytoplasm of that host cell tells the genes, hey, switch off, stop misbehaving, and stop causing cancer. This means that genes are switched on and off by signals from inside or outside the cell. So that is where the message is coming from. The message is saying to us, the genes are at the mercy of commands from the cytoplasm of your cell. Cancer is caused not just by mutations, but by merely cytoplasm that sends out the wrong signals. Okay. But my Professor Vilan Givas, when he saw all these books, he was a bit worried because he qualified in the time when the gene theory was dominant. So he says these books are fine, but they have minimized exploration of the deep ways that highly evolved genomes ensure that every newly reproduced body conforms to the essential features. And he made statements like, if you go out into the, say, into the say to Serengeti, and you see a herd of buffalo, you won't be able to recognize one buffalo from the other because the genes gave the buffaloes the identical plan. You'll have to go very close and take two buffalo and say, this one's eye, the left eye is a bit larger, the other one has got a left eye that's smaller. So he says this is a genomic blueprint. So this debate is on, and it's an extremely exciting debate. Right? But this is the problem. He says the books are fine. The only real problem with Philip Ball's fine book is thus the repeatedly stated view that the genes contribute, but by no means determine the outcome of organismal development. He doesn't like that statement. So I'm having an exciting time with him. I visit him, we have lunch, and we have these debates, okay? And if you want to read his review, it's in the South African Journal of Science, 2024. You can just Google Philan Givas. Ladies and gentlemen, what Siddhartha is telling us in The Song of the Cell, which is one of the most fascinating books, Siddhartha Mukherjee, he said genes by themselves are strikingly incomplete explanations of the complexities and diversities of organisms. We need to add gene-to-gene interactions and gene-environment interactions to explain our physiology and our fate. Also, cells by themselves are incomplete explanations of organismal complexities. Again, we need to usher in interactions between cells and their environment. And I used to discuss this with my late cat, Mika Mall, but he's not here. So today I've decided to discuss it with you. Thank you very much. I think we've made it on time.
Thank you very much, Professor. And that was really an exciting talk,
Thank
interesting
you.
information. I'm not sure if there is anything on the chat because you clearly explained everything very clear. And it's actually like a new or amazing point of view of the origin of life, basically.
Yeah. Well, thank you. Can you ask the audience if there are any questions?
Yes, I can see
Yes,
Tedmer.
yeah, I have a question.
Yeah.
That was an excellent overview. Thank you so much.
Thank
Really,
you.
I
Thank you.
learned a lot, but at the same time, I'm confused a lot
Okay.
about the life. I don't know where to start and where to end. So it's everybody hypothesized different ways as you outlined, but I don't know where the truth lies in terms
Okay.
of origin of life. So it remains for me. The truth is unclear, except
Right.
believing God creates life. That's what I think right now from my end. But in terms of the history, I feel some of the key players, probably like Fisher or others, were not mentioned.
Right.
I don't know whether their contribution is not relevant from your perspective, because those are the changing moments.
Right.
So
Right.
my last point is, in those many centuries years, there's no much development in, at least in science, but the last 10, 20 years, at least in human genetics, we see big data unfolding all these different, areas. Do you think this change will at least help us to understand in a very short time the points you mentioned, or we just still debating as much as we can?
Right.
So,
So
things, but sorry, it's kind of different things I tried to mention, but
Yeah, no, I appreciate it. I didn't hear you clearly. Did you say from your side, you say God created life?
Yeah, that the hypothesis you described to me, different individuals
Yeah,
come from different sides, and I don't know which one to believe because it's a hypothesis. There's no proof.
right, right. So that's the creationist view, and it's a very strong view. I grew up with that view. Uh, and then there's the scientific view, which is a different one. And so when we sort of when scientific activity becomes our professions, then we try and look at this from a scientific point of view and the evidence-based view. But there's nothing wrong in holding both views together. But there are people who hold the creationist view entirely. Some people who, like Richard Dawkins, who's a militant atheist, says, I hold the scientific view because it's evidence-based. So I've got no disagreement with you on that point. The other point is that you are right. You're mentioning Fisher and Company. But what I've done is, for a 40-odd minute talk, I've taken the most recent debate by two people. Dawkins, who came out of the information theory and published that book, and he still holds that the genes are the most important role players here. And then there's Nick Lane, who came later from University College London, who said, listen, we're being too narrow-minded. Our outlook is too narrow. We need to talk about life, its origin, and then find out how it could have originated and what it means to be alive. And so he formulated this theory. Now, can you see the slide? This slide I've got on
Yes.
now.
Yes,
Right.
yes,
So now,
yes.
when they say we're made of atoms which make molecules and then cells and then go up that way, so there are people who say that the arrow points all the way down. That means there are many scientists who believe, why are we so worried about all this complexity? We should just know that atoms and chemistry and physics gave us all the matter in the world, including the living matter. Now, that is called reductionism. That means if I took you, a live person, and broke you down to your most simple units, there'll be atoms. And if I took the table I'm sitting at and broke it down, there'll be atoms. And they say that is very important, right? But now this new theory is saying, no, 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 no. The genome by itself only acts on instruction from the outside. That means from inside the cell or from outside the cell. So we can't only say the arrow points all the way down, because as we've increased in complexity, new properties emerged at each one of these very creative levels. And when you get to a level, you will never be able to predict where it came from. And if you're at a level, you will never predict where it's going to. This is the whole fascinating point about it. So they say if you want to actually define life, it's still unclear, you are right. You have to consider every step here and not just say it's atomic and we're made of atoms, full stop, or atoms made molecules and chemistry and we are a bag of chemical reactions. Those are what they call reductionist statements. What these people are talking about is the holistic scientific view that things happen from here, go up, and once this happens, this affects you here. Like if you suddenly saw a horrible accident, you as a human being, you conscious human being, you saw a horrible accident, you have changes in your body because what you saw goes from your mind right down into your cells. And you might get the cell producing some coding for some kind of hormone like cortisol, a stress hormone to calm you down. So life includes, embraces all these levels and goes up this way and goes up that way. And that's the scientific view. And you're right, there are many more contributors to this. I agree with you, and I'm glad you raised that. Thank you very much. Any other questions?
Thank you, Ted.
Thank you very much. I don't want to dominate, but I have the last question I mentioned was the scientific progress in the last 20 years versus the time before. Do you think recent big data and other will change the way we think life in the future?
Yes, I think so. If you consider that Darwin formulated his theory in 1859, right? Einstein talked about atoms for the first time in 1905. So Darwin came in 1859, and we only found the unit of hereditary, which he didn't know about, which he suspected existed, didn't know about, 160 years later. And now suddenly we've mapped the human genome, and from there, so much is happening. As you say, the data is, we've got more modern equipment, we've got fresher young minds, we've got, well, bigger labs, more money being put into the sciences in places like the Northern Hemisphere. We're just hoping that things change on an international level, that the Southern Hemisphere starts benefiting from a lot of this. And I think that things are being concentrated. They're happening very fast within shorter spaces of time. So I think, like, if I'm an old man now. If I lived for another 10 years, I'll be quite shocked at the amount of progress that's made in those 10 years, because I'm very shocked at the amount of progress being made since the human genome mapping and today. Yeah, so I believe that it's becoming, the growth of knowledge is accelerating. Thank you.
Yeah. Thank you,
Thank
Prof.
you very much. Long live.
Thank you.
Thank you very much. I see many thank you messages on the chat, and I think we finished a little bit over time, but it's been an honor to have you in, and that was very amazing and interesting talk. Thank you, everyone.
Okay.
Any further question or anything, feel free to pop it up in the to pop an email to our society.
And
Yeah.
Salma, feel free to share my email address if people want to continue the discussion, because I just love it, because I learn a lot from these discussions too. Thank you.
Let me just put it now if anyone wants.
Yeah, it would be good if you put in the chat box.
Yeah.
Fantastic.
There you go. Thank you very much, Professor, for your time and for your talk.
Thank you.
And
And thank you all for attending. I really enjoy that.
Yeah. We'll
Thank
see you, everyone, next month.
Bye-bye.
Thank you. Bye.